Hi everyone, I am Dr. M. Parimala, Assistant Professor, PG and Research Department of Microbiology, St. Joseph's College of Arts and Science, Autonomous Cuddalore, Tamil Nadu. I am going to present a topic enzyme immobilization. Now we will see the introduction. What is enzyme immobilization? Enzyme immobilization may be defined as a process of confining the enzyme molecules to a solid support over which a substrate is passed and it is converted to product. Actually, this is the production process, that is, we are producing a product using a substrate. For this, an enzyme is necessary. That enzyme has to be immobilized in a solid support so we can reuse it again and again. And regarding in this process, the stability is found to be an important criteria, as well as the physical forces that is existing in between the carrier molecules. So this is called the enzyme immobilization process. And the immobilized enzyme is the one whose movement in space has been restricted either completely or to a small limited region. So we are immobilizing, that is, we are restricting the movement of the enzyme so that we can maintain the enzyme in a fixed place. So we can add the substrate and the product production can be continuously made. So this will be it will be carried out in a restricted area or to a limited area. And now we will see what is the need for enzyme immobilization. First one, protection from degradation and deactivation. If we are using the enzymes, once we are used, it will act upon the substrate and produce the product. So in this, if you are immobilizing the enzyme, we can maintain that for a longer duration, as well as we can protect them from degradation and deactivation while it will be attached to a solid support. Then reuse of these enzymes for many reactions. So this is the major application. Thus we are lowering the total production cost of this enzyme mediated reaction. So if we are using again and again the immobilized enzyme for the production process, we are reducing the cost of that particular enzyme for production process. And also the ability to stop the reaction rapidly by removing the enzyme from the reaction solution. So in this, if you are carrying out a reaction and immediately if you want to stop the reaction, we can remove the immobilized enzyme so that the reaction can be stopped immediately. And next point, enhance stability. While attaching the enzyme to a matrix, automatically we are enhancing the stability of the particular enzyme so that we can reuse it again and again. And next one, easy separation of the enzyme from the product. So since the enzyme is immobilized, so while separating the product, it is very easy to separate the enzyme. So this is a beneficial one. And the product is not contaminated with the enzyme from the product. So if the main objective is to separate the product. So while the separation of this product, you can easily separate the enzyme since it is immobilized in a matrix solid support. So the product recovery is found to be very easy. And now we will see the properties of the carrier matrices that is used for enzyme immobilization, because we have to immobilize the enzyme by add this to a carrier matrix. So the carrier matrix should be, first thing is inert, that is it should not give any reaction, so it should be an inert one. Next one, it is physically strong and stable so that only this enzyme can be immobilized using that substrate. Then third point, the cost wise, cost effective. So if the cost of that particular matrix or support is found to be high, it is not economical. So cost wise we have to consider. So if the cost is found to be less, it is found to be more economical. Then the fourth one, regenerable. So if it is regenerable, then it will be well and good. Then the last one, reduction in product inhibition. So the main objective of this using enzyme is to produce the product. So it should not react indirectly by inhibiting the product formation. So all these criteria has to be well satisfied by the matrix, that is the carrier matrix that can be used for enzyme immobilization. And in this slide, you can see the methods that is used for enzyme immobilization. Actually, two methods are generally followed. First one, physical method, and the next method is the chemical method. So we will see what are those physical methods: adsorption, entrapment, and encapsulation. So these three methods, they are coming under the physical method, and under the chemical method, covalent binding and cross-linking methods. So totally these five methods are commonly involved in the considered as the methods for enzyme immobilization. The first method, adsorption, as the name indicates, it shows adsorption over a support. Physical binding of this enzyme on a surface of a carrier matrix. So we are using a carrier matrix. Over the carrier matrix, this enzyme has to adsorb. So the carrier may be either organic or inorganic. So both the choices are there. And the process of adsorption, it involves the weak interactions like van der Waals force or hydrogen bonding. So weak interaction will be noted in between the enzyme as well as the supporting matrix. Best example for the carriers that is used for adsorption are silica, bentonite, cellulose, etc. And the enzymes which is used for such that can be immobilized by using this adsorption process are catalase and invertase. You can see in this picture the adsorption of this enzyme molecules over the matrix, that is the carrier matrix. And in this method, it is having both advantages as well as the disadvantages. First, we will see the advantages. It is found to be very simple as well as economical and limited loss of activity. So the activity will be prolonged. So it is found to be well and good. Then it can be recycled. Then it can be regenerated and it can be reused. So we can use this immobilized enzyme for a longer duration. So it is a major advantage. And regarding the disadvantages of this adsorption method, it is relatively low surface area for binding. The surface area that can be used for adsorption is found to be less when compared to other methods, and exposure of this enzyme to microbial attack will be more. So it is considered as the disadvantage. Also, the yields are often low due to the inactivation and desorption. So it is found to be it is considered as the major disadvantage. And now we'll see the next method, entrapment. In entrapment, the enzymes or the cells are not directly attached to the support surface, that what we have seen in the adsorption, but simply it is trapped inside the polymer matrix. So in this method, we are using a polymer matrix into which these enzymes are entrapped. So it is called entrapment method. The enzymes are held or entrapped within the suitable gels or fibers. So in this method, we are using two components. One is the gel and another one is the fiber. If it is done in such a way to retain the protein while allowing penetration of substrate, it can be classified into lattice and microcapsule types. And now we will see how this can be entrapped in gels and fibers and with examples. if we are using the gels, that is inclusion in gels, there are certain substances can be used, that is polyacrylamide gel, polyvinyl alcohol gels, etc. And if you want to make inclusion in fiber, we can use cellulose. Then if you want to include in microcapsule, you can use polyamines, polybasic acid chloride monomers. So this in this picture you can see very clearly how this enzyme molecules get trapped into the fibers as well as the gels. And in this method you can see the lattice type of entrapment. It is one type of entrapment, that is the enzymes will be entrapped in a matrix. Entrapment involves entrapping this enzyme within the interstitial spaces of a cross-linked water-insoluble polymer. So in this we are using a polymer that is found to be water-insoluble, and we are introducing the enzymes in the interstitial spaces. Some synthetic polymers that can be used are polyacrylamide, polyvinyl alcohol, and some natural polymers like starch that can be used to immobilize the enzyme using this technique. So this is called lattice type of entrapment. So this enzymes will be added to the polymer matrix and that will be found within the interstitial spaces. So that you can see very clearly in this picture. And the next method is encapsulation. The term itself shows it will be enclosed by a capsule-like structure. So it includes enclosing the enzyme with a semi-permeable polymer membrane. So for this encapsulation process, we are using semi-permeable polymer membrane. Example, semi-permeable colloidal or nylon membranes in the shape of sphere. So we are encapsulating in this picture, you can see very clearly we can encapsulate this enzyme using a spear shaped colloidal structure. So this method is said to be encapsulation. And we will see the advantages as well as the disadvantages of this method. Here there is no chemical modification. Relatively it is a stable form and easily handling and reusage. So you can handle this very easily and we can reuse it for a longer duration. So it is an advantageous method. And if you consider the disadvantage, sometimes the enzyme may leak from the pores. So it is considered as one of the disadvantage associated with this method. And now we will see the next method, covalent binding. Based on the binding of enzymes and water insoluble carriers by covalent bonding. Covalent bonding is found to be a strong binding that is associated in between the enzyme as well as the carrier. The functional groups which may be actually take part in this binding are amino group, carboxyl group, sulfidyl group, hydroxyl group, imidazole group, phenolic group and thiol group. And while considering the advantages and disadvantages, it is having more advantage. The binding force between the enzyme and the carrier is said to be so strong when compared to other methods. And there is no leakage of enzymes that occurs in the other methods, even in the presence of the substrate or a solution of high ionic strength. So if you are using a substrate with a high ionic strength, even at that time, the bonding is found to be so strong, so it can be having the more advantageous than other methods. Then while considering the disadvantage, sometimes it may alter the conformational structure and active center of the enzyme. So this may occur at sometimes, so which results in the loss of activity, so the production may affect. So the limited disadvantage may be seen along with this method, that is the covalent binding. And now we will see the next method, cross-linking. This cross-linking involves intermolecular cross-linking of enzyme molecules in the presence or absence of solid support. So we are using a solid support. In this, the enzyme will be linked. It will be linked to the solid support. This method produces a three-dimensional cross-linked enzyme aggregate. So the production of this enzyme substrate complex resembles like a 3D cross-linked enzyme aggregate. So this is mainly due to its insolubility in water by means of the multifunctional reagent that links covalently to the enzyme molecules. So we can produce the cross-linked enzyme, so that is found to be very useful in the production of the product. And we will see the advantages of cross-linking. Very little desorption, that is the enzyme will be strongly bound, so it is difficult to separate out, and it is having higher stability. The stability of the molecule is found to be high, even though there will be variation in pH or ionic strength or substrate concentration. So whatever may be, it is found to be highly stable. And while considering the disadvantages of this cross-linking, the cross-linking may cause significant changes in the active site. So actually for enzymatic reaction, active site is found to be important. So if there is any modification or changes in the active site, it is not considered as a safer one and also not cost effective. So cost wise also it is not good. In this picture, you can see the 5 different methods that what we have discussed in the previous slides. Adsorption, cross-linking, covalent bonding, encapsulation, and cross-linking. So these 5 methods of immobilization are commonly used for the enzyme production, that is the enzyme usage in the production of a product using the substrate. And now we will see the limitations of enzyme immobilization. The cost of carriers and immobilization is found to be a major criteria. So the cost wise we have to consider. Next one, changes in properties, that is selectivity. So selectivity is also very important. If there is any modification or changes in the selectivity, it is not good. Then mass transfer limitations are there, problems with cofactor and regeneration is there, and also problems with multi-enzyme systems are there, and also activity loss during immobilization may be also play a major role. So some of the limitations are there, even though the use of immobilized enzyme, it is found to be beneficial cost wise and also stability wise. So we can produce the product for a continuous process using the substrate. Thank you.